evening. My name is Anne Ophelis, broadcasting the 7 o'clock evening news live from London. So tonight, malaria continues to be a massive global threat. We will shortly be talking with the chief executive of a growing pharmaceutical company, chloelimxiuyu. But first, malaria is a life-threatening disease with over 290 million cases reported in 2017. Of those cases, 425,000 lead to malaria deaths. The World Health Organization estimates that 92% of malaria deaths and 93% of cases relate to the African region, an area of the world that does not have the financial backing to combat this deadly disease. Most shocking of all, 70% of malaria deaths occurs in children under the age of 5. That's one child every 2 minutes. This is why chloelimxiuyu are on a mission to provide new, effective anti-malarial treatment. We are now joined by Jamie Burrows, Chief Executive of chloelimxiuyu.
So thank you for having me.
Okay, so firstly, what are you aiming to achieve from this project?
So we are aiming to gain financial investment from investors so that we can carry out the synthesis of potential new effective drug candidates, and also to run bioassays in order to test the effectiveness and the toxicity and safety profiles of these drugs. So the overall objective is to come up with a more cost-effective drug candidate that can tackle the resistance problem seen with many of the current antimalarials, such as chloroquine.
So why is resistance such a problem?
So the widespread use of antimalarials has led to the development of Plasmodium falciparum resistant strains in almost all areas where malaria is transmitted. This figure shows the extent of the resistance. So the white dots represent the resistant strains which can be seen across Africa, South America and South Asia. It's one of the biggest challenges facing malaria control and highlights the importance of the development of novel antimalarials.
Thank you, Jamie. We now go to Somaya Alshik, the Chief Medicinal Officer of chloelimxiuyu.
Hi Anne, thank you for having me today.
So, hi Somaya, could you tell me more about the resistance of malaria?
Yeah. So in order to understand the resistance of malaria, we need to understand the current mode of action of the medicine that's currently available in the market, and that medicine is called chloroquine. So chloroquine works by inhibiting the polymerization of hematin to hemazoin, causing a buildup of toxic heat in the parasite, which leads to parasite death. But in order for chloroquine to do that, it needs access to the food vacuole in the cell. So it moves down a pH gradient from basic to an acidic environment, and this causes the quinoline and tertiary amine nitrogen of the drug to become protonated, and that would cause an accumulation of the drug in the food vacuole. The continuous use of chloroquine has led to the development of resistance in more than 80% of malaria strains. It's still a developing area of research, but current research proposes that resistance is likely to be due to mutation in the transporter gene B04. The main role of the transport is the transport of amino acids and small peptides from the hemoglobin degradation outside the food vacuole. The mutation repulses the diprotonated chloroquine from the food vacuole to outside the food vacuole, and which results in a decrease in the drug concentration in the food vacuole and no drug action.
Okay, so could you tell me more about the compound that you used to come up with the new antimalarial?
Yeah, so the compound that we used, as you can see on the diagram, has a lot of similarities between it and chloroquine. It basically has the essential groups for the basic activity of the drug molecule. So for example, the tertiary amine highlighted in dark blue is important for the accumulation of the drug in the food vacuole by forming an ionic interaction with both the oxygen atoms of the carboxylic acid on the hematin group. And also, research shows that the lead compound shows activity in both chloroquine-resistant strains and non-resistant strains. So that's a really good sign. Okay, thank you.
Thanks Somaya for the explanation. We have also invited Chloe, the research and development chemist of chloelimxiuyu.
Thanks for the invitation, Anne. It's my pleasure to speak here.
Okay, so Chloe, what has the company done to make the lead compound better?
Okay, so there are a total of 28 compounds that we came up with after considering their pKa profiles and the oral bioavailability by undergoing the pKa analysis and the drug likeness analysis. We aim to reduce the pKa of both quinoline and nitrogen, and also the basic nitrogen, to improve the drug activity. Also to make sure that all our compounds are predicted to be orally bioavailable by taking into account that the Limxiuyu rule and the Weber's rule. Now we are very pleased to say that all our compounds can potentially show a good degree of activity. In terms of lead optimization, we look into a total of 4 sites of our lead compound to improve the anti-malaria activity against the chloroquine-sensitive and the resistant strains of the P. falciparum. First of all, we replace the chlorine at the 7th position with a benzene ring to increase the lithophilicity of the compound. Next, we also modified the quinolone ring by adding the methoxy and the chlorobenzyl group. Furthermore, we modified the length of the nitrogen linker chain. A benzene ring was added in between the linker chain to increase the lithophilicity of the compound. Moreover, we modified the side chain of the basic nitrogen, such as the substitution with a nitrogen third butyl group, the introduction of the phyrolysidinyl moiety at the pendant nitrogen, the addition of a second tertiary amine, and the introduction of the pyrrolidine group. All of the modifications can potentially tackle drug resistance, and these are actually supported by evidences from relevant resources available.
Thank you, Chloe. We are now joined by Lynn, a medicinal chemist who is reporting from the lab about the synthesis and metabolism of the drug.
So we aim to run parallel synthesis of our 4 aminoquinoline analogs using expeditious synthetic routes, which are also highly cost-effective. That way, we'll be producing compounds at high yield and at a level of purity that is required for biological evaluation. Our principal synthetic approach is direct and regioselective substitution between nucleophilic diamines, which are our newly designed side chains, and a range of haloarenes, which are the 4 modified quinoline cores. Because most of our starting materials are readily available from reputable sources worldwide, our synthesis project can start immediately once we have received the grant. As part of preclinical study, we've also predicted the metabolic pathways that our compounds can undergo once enter the body. The analogs can be extensively metabolized into more polar and water-soluble species, which will then be easily excreted. A few exceptions are those shown on the screen, which were intentionally designed to resist biotransformation and therefore enhance anti-malaria activity, especially in resistant parasites. We've also identified a few potentially toxic metabolic intermediates. However, by having optimal target binding affinity, we are certain that this dose-dependent toxicity will not cause major issues.
Thank you, Lynn. We are now joined by Mira, the Chief Operating Officer of Quinoceuticals. So how are you going to test whether these compounds are safe for people to take?
Okay, so we're very pleased with the compounds that we've designed, and we're going to run 3 bioassays on our project. So bioassays are laboratory tests which we run to test how safe and effective drugs actually are. The first assay that we're going to use is the beta-hemotin inhibitory assay, which allows us to test against Plasmodium falciparum so we can identify resistance early on. The next assay is the hypoxanthine incorporation assay, which allows us to calculate the IC50, showing us how potent our compounds actually are. It allows safe and effective doses to be estimated for patients, and we estimate that these assays will take around 2 to 3 months, which includes the lab side and the analysis. The next assay that we'll use is an in vivo study, which is going to be tested on mice, and we test the compounds on these mice that are infected with the rodent strain of malaria cells called Plasmodium burkii. The compounds will be compared to a known anti-malarial as well as no drug to identify how effective they are, and this process is estimated to take around 6 months, and any successful compounds then go on to phase 1 of clinical trials.
So how long is the project expected to take?
So obtaining our starting materials and commercially available products is expected to take about a month, and then synthesizing our analogues is estimated to take around 3 months. So the whole project is estimated to take around 14 months, which will be overseen by a project manager.
Alright, thank you, Mira.
Thank you.
Compounds have the serious potential to combat the anti-malarial resistance problem. We hope you will not only invest in our project, but invest in a future where malaria is no more.
Let's work together to make malaria no more.